Okay, lab number 10 as well as a lab practical review. Lab number 10, angiosperms, angiosperms, flower and seeds. And today we'll talk about the fruits. And remember, we have two different major types of the fruits, the fleshy one and the dry, relatively easy to identify. And I'll outline what exactly needs to be done in order to successfully answer the questions on the lab practical, which will be, by the way, on Tuesday, right? After the lecture, we'll have a lecture, and then I think we'll go into the lab practical, and I'll address a few issues with the lab practical during that lecture. Anyway, pre-lab questions. What is the reproductive organ of angiosperms? Flowers. Ta-da, that's the reproductive organ. What two parts make non-essential parts? Non-essential parts, the one which is not producing male or female gametes. So we're talking about the sepals, or the whole collection of the sepals known as the calyx, and then the petals, and that known as the corolla, all of the petals in the flower. What do we call male reproductive structure of the flower? And here we have it right here. So that would be anther, right? With a filament overall. I think it says right here. Yes, stamen. But if you'll answer anther, I'll be okay with it. What is the name for the part of male reproductive structure that bears pollen? Now we are specifying, and it's anther, right? What do we call female reproductive organ or structure of the flower? It could be a carpel, C-A-R-P-E-L, or pistil. It's the same thing, essentially same thing. So either or will do. Some students like, what about carpel? I'm like, yeah, it's the same thing. Okay, so don't get confused. Where is the egg found within an ovule? Within an ovule, it will be located. So we have ovule, and the ovule located in the ovary. And remember, overgrown ovary generally become fruit. What is another name for the female gametophyte? Megaspore, right? So I think we have it somewhere here. Meiosis, yes. Ovary, and within the ovary we have the megaspore. What is the diploid structure found as a result of fertilization? That would be a zygote. Explain fertilization, why fertilization in angiosperms called double fertilization. And that's a question on the test, so make sure you understand that. So we have a haploid in the flower, we have a haploid sperm, we have two of those. So 1 and 2, right? So we have two sperms. So one sperm goes and fertilizing the egg right here, the egg right here. Oops. Fertilizing the egg, creating a diploid zygote, which will develop into the embryo. That's the first round of fertilization. But we have a second sperm. Second sperm comes in, the egg no longer can be fertilized. So what it does is fertilizing the central cell, right? And central cell is dikaryotic. Whenever the sperm comes in, it creates three nuclei which merge together, creating a triploid, 3N structure, and that will develop into an endosperm, food source for the embryo. So as a result of double fertilization, we have two different cells being fertilized by two different sperms, one sperm fertilizing the egg, another sperm fertilizing the central cell. The egg fertilization will result in a zygote, which will develop, diploid zygote, which will develop into the embryo, and the central cell fertilization will lead into a triploid structure, which will lead into an endosperm, or will develop into endosperm food source for the embryo, developing embryo. All right, so now table 1. Whenever you're looking at the table 1, you see the model of the flower, and again we're looking at the green part, which is the calyx. Then we're looking at the colorful part petals, and then the next wall will have the stamens, right, with anthers and the filaments, and then the central part is actually a pistil. So in this particular case, receptacle is a part where the ovary sitting on, so to speak, or located on. So here we have receptacles, sepals are the green, petals are, well, here it's kind of, I don't know what the color will be, orangish, light orange, I guess. So petals, stamens, that particular, the white filament and the yellow anther. So anther, a yellow, white filament, pistil, the whole this structure. And within a pistil, we will recognize the top part known as a stigma, the elongated part known as a style, and that round part known as the ovary right here. All right, and in this particular case, the ovary contains only one ovule with only one egg. Have this flower been pollinated? Whenever you're looking from the top, and I specifically took the picture looking from the top, you'll see those orange dots, right? Orange dots are pollen, so pollen already landed on stigma. So yes, it has been pollinated. Have it been fertilized? Let me so here we go. This is the pollen tube, the orange, and within the pollen tube you have 1 and 2 tiny little bitty brown dots. That's the sperms. We have 2 sperms. Here we're looking at the red cell, that's the egg, and that one is the central cell. It's literally in the center. So that particular flower has been pollinated, but the sperm have not reached the egg, so it have not been fertilized yet. Therefore, no, but literally within a few seconds, the sperm will, well, in the real flower, the sperm will merge with the egg, and that would be creating a diploid zygote. And then the second sperm will go and merge with the central cell. So here we go. Here it's on that picture, you can clearly see 2 of those dots right here again. All right, so label ovule, integument, embryo, sac, egg, synergetic cells, polar nuclei, antipodal cell. All right, antipodal cell, they will be on the opposite side of the egg. So egg is a red right here. So antipodal cell on the opposite side, anti, against, right? So here is antipodal cell. Then the red one would be the egg. Right next to the egg, there is 2 cells which known as the synergid cells right here. The polar nuclei. Polar nuclei or central nuclei, that would be right here. That central part, 11, I guess. Then the embryo sac. The embryo, it's kind of hard to, so all of that is the ovule, right? Within the ovule, we have the embryo sac. The integument would be the outer part. The whole thing is the ovary. So ovule is that particular circle, kind of. But again, on the lab practical, it would be really hard to label that, but you definitely need to know central cell, the egg, the sperm, so it might point it out to those particular parts. Which cell will combine to make up the endosperm? The central cell and the sperm. Is the endosperm haploid, diploid, or triploid? The central cell has 2 nuclei, also known as the polar nuclei, right? Because they're kind of on the opposite poles of the central cell. And then we're adding another nuclei from the sperm. Each one of them are haploid, so 1N, 2N, 3N. So it will be a triploid. Which cell will combine to form a zygote? Sperm and egg. Is the zygote haploid or diploid? Haploid sperm, haploid egg combined together will end up being a diploid structure. Why it's called double fertilization? We already went over that, because there is 2 merging, well, 2 sperms fertilizing 2 different cells. the egg and the central cell, resulting in the zygote, diploid zygote forming the embryo, and triploid endosperm forming the food source for the embryo. All right, any questions on that? No questions. All right, so here we're looking at the non-pollinated and pollinated pistil, right? Well, technically flower, but it's you can see the on the stigma the pollen. All right, so here we have antipodal cell, polar nuclei, synergetic cells, and the egg cells. Those ones you will need to know for sure. So again, the picture, receptacles already kind of cover that. Now, flesh flowers. On the flesh flowers, remember, two very, well, whenever you're looking at any kind of flower, you're first counting the petals, the easiest one. Sometimes you can count the anthers as well, but also make sure that to take a look on the leaves. Quite often, whenever you're looking at the flowers, the leaves will be available. So whenever you're looking, in order to identify if it's a monocot or dicot, generally, monocot flower will have a multiples of 3, like petals 3, 6, 9, and so on. And for the dicot, it would be 4 or 5. And I'll show you the flower, and I'll talk to you about a little story. So anyway, looking at this particular one, you will need to label petals, stamens, and pistils. On the lilies, you don't really see the sepals, but you can see clearly the bright yellow petals, the filaments carrying the anther stamens, and then of course the central part would be the pistil. So in this particular case, the petals, stamens, and that one is a pistil, including the ovary. All right, so here you're looking at that and you're counting 1, 2, 3, 4, 5 petals. That's clearly a monocot, right? Why many flowers are so colorful? To attract the pollinators. We already talked about that. Did any of the flowers examined today missing the non-essential organs? That particular one, the yellow lily, missing what? Sepals. Does not have sepals. Has the petals, but not sepals. Did any flowers examined today miss any essential organs? By essential organs, we're talking about anthers or pistils, and no, I don't think we did. So here, let's take a look. Monocot or dicot? Type it in, let me know, what do you think? That's a crocus. In the, well, usually they grow early in the spring. So monocot, dicot. You should be able to identify just looking at the picture. Because on the lab practical, you're coming in, there might be a picture and it says which group it belongs to. And you're like counting 1, 2, 3, 4, 5, 6 petals. 6 petals will be a monocot. Yes, good idea. Let's take a look. What about this guy? Oh, those guys. You can clearly count 1, 2, 3, 4, 5 petals. So 5 petals right here. Whenever you're looking at this, 1 petal, 2 petals, 3, 4, and 5. I had a student before coming to me in a face-to-face class with that particular flower and saying, well, I'm not really sure if it's a monocot or dicot. I'm like, why not? And he's like, well, the number of the petals in the flower, and he's showing me the flower, is 7. 7 is not multiples of 4, not multiples of 5, not multiples of 3, of course. So I'm like, well, what do you think? Is it a monocot or dicot? He's like, I don't know. It's a petals. It's a 7 petals. It's not fitting into the any of the rules. I'm like, okay, well, let's take a look on another flower and also look at the venation. Here you have a pinnate venation, so non-parallel, most likely a dicot. And now let's take a look on another flower. And we looked at another flower and it had 5 petals. He's like, what about this one? I'm like, well, that's a mutant. It had a 7 petals. sometimes happens. So careful with that, all right? So those ones, you're looking at the dicot. What about this guy right here? The guy on the left. Monocot, dicot. Dicot, how do you know? Kind of hard to count the stamens. There is pretty much no petals. Stamens are actually showing off the flower. But in this particular case, again, you're looking at the leaves, and the leaves showing the pinnate. What about this guy, which goes in my front yard? That's literally a picture from my front yard. Well, that particular one, last spring. No, spring before, because last spring it did die. Well, it didn't die, but it froze. That's a monocot, yes. 6 petals right there. 1, 2, 3, 4, 5, 6. So be careful. There will be questions from those particular pictures, obvious, easy to count, and it will say name the group of the plants. Let's say, do I have? No, nothing else. All right, now fruits. This is getting a little bit confusing because we didn't spend too much time, and I don't have that time to go deep into the fruits. And basically, in this particular case, the easiest way for you to prepare yourself, and there will be some fruits, but it will be again several questions on the lab practical, is just to memorize what we have for the samples. Later on, if you'll go into a botany, you will describe, well, you will discuss with your professor what makes pepo a pepo, for example, and what's make a hesperidium. But it's everything to do with the pericarp and the carp and so on. So here's the major ones. I would not be looking for these ones. I don't really worry about those ones. So whenever you're coming on a lab practical, you need to identify several things. First of all, if it's a fleshy or dry. If it's a fleshy, you will have several options. If it's a simple fruit, if it's an aggregate fruit, or if it's a multiple fruits. Okay, that's for the fleshy. So if you'll see the picture of the fruit on a lab practical, first thing which you need to identify, fleshy or dry. If it's a fleshy, is it a simple? Is it the aggregate? Is it a multiple? I don't care about pepo, hesperidium, berry, or anything like that. Again, didn't have the time to go over why the berry is the berry, why the palm is the palm. So in this particular case, I will not be asking, but I will be asking simple, aggregate, or multiple. That's if it's a fleshy. If it's a dry, it's already by the definition technically a simple one. Then you will need to figure it out if it's a dehiscent or indehiscent. Again, just refreshing your memory from the last time, dehiscent fruits, dry fruits. Whenever the fruit is ready, it will literally open up, releasing the seed. If it's an indehiscent, it will stay closed, and whenever the embryo grows, the seedlings grow, it will have to break its way through. Okay, so essentially we're looking at the 5 different types of the fruits. We're looking at the fleshy simple, fleshy aggregate, fleshy multiple, or dry dehiscent, dry indehiscent. Make sense? All right, so let's kind of take a look on what we have on the table, and we'll fruit name. Tomato. Okay, so is it a fruity or fleshy or dry? That's the fleshy. Simple or compound? That would be a simple one. Just, and again, what's the difference between simple versus multiple? 1 flower and 1 or more, well, 1 ovary. 1 or more ovules. As a result, you might end up with 1 seed or multiple seeds, but that particular one is the, well, that's a tomato, fruity, simple, and type is a berry. You don't really have to worry about it, but yeah, here we go. Another one, fleshy, simple. Well, that's a grape, of course. So grape, fleshy, simple, and the type would be a berry again. So here what we have, berry. But again, on the lab practical, if you'll say me, it's the simple, fleshy, simple, I'll be okay with it. If it's a simple, oh, not simple. If it's the compound or fleshy and then aggregate, I'll be okay with it as well. So next one, that's lime. And lime is again fleshy, simple, and the type of the fruit known as the Hesperidium. I think we have it right here, yes. That's the type of the fruit. So here we go. So as the oranges and the tangerines and the pomelos and grapefruit and so on, all of the citruses, oils, well, lemons as well. All right. So next one, here we have a peach, not peach, plum. So we have plum, and you can see one seed only. If it's a one seed only, it's automatically simple. Fleshy, simple, and in this particular case, so same thing here, we're looking at the peach, one seed only, it's automatically simple, fleshy. And anybody knows what's that? That's something which known as the pluots. What the pluot is? It's a mix, it's a hybrid between plum and apricot. Combined together, created a pluot. So, and those ones, they known as the drupes, but you don't have to worry about the drupe part. Peach, the pluots, apricots. So we'll have a hard coat, the stone peak. Peaches, plums, all that stuff. All right. Now here we're looking at the cucumber. And that would be the pepple. So it's a simple fleshy fruit, and then the type known as the pepple. Apple. Apple is a little bit, technically it's an accessory fruit in the simplicity, and we haven't talked about the accessory fruit. So for the simplicity's sake, we will keep it as the simple fleshy fruit, which is technically wrong, and yes, botanists will crucify me if I'll just say it's a simple fruit. It's not, it's accessory fruit, but we're not going on that level of botany, we're not going too deep into that. So apple will consider it as a fleshy, simple, moving on. All right. And it has a special type pom, well, yeah. Now we're going to the aggregate fruits. So here we're looking at the raspberry, blackberry. So raspberry in this particular case, it's a single flower, but each one of those parts created by the separate ovaries. So it's the ovaries fused together. So multiple ovaries known as the aggregate. Raspberry would be one of those particular good examples. Strawberry is also can be considered as that. Again, it's a little bit more complex than that, but we'll keep it for the simplicity's sake. It's a simple. No, it's not simple. It's a flashy and it's an aggregate. All of those particular seeds has been created by different ovaries. They were allocated in the different ovaries, which later on fused up into a fruit. Again, one of the accessory fruits, but again, we will, if you'll see the strawberry on the lab practical, says flashy, aggregate, move on. You'll get the full credit. Here we're looking at the blackberries, same thing, flashy, aggregate, move on. So here what we technically looking aggregate, one flower, but it's creating tons of different, well, it's made out of tons of fused up small ovaries. Pineapple will be something which known as a multiple fruits. Essentially, each one of those particular kind of squarish or rectangles, I guess it's like a diamonds. That's a single fruit of the pineapple, which are fused together with the several one. Therefore, it's kind of multiple fruits fused together, creating that particular thing. So pineapple is a great example, and I think it's probably the only example I have for the pictures. Yes, the only one I have for the pictures. Figs are another one, but I didn't take the picture, so it probably will not be there. And the mulberry, I need to go, and I know the place where they grow wild, semi-wild in not far from me, but yeah, later on, maybe at some point of time. So here we go. If you'll see pineapple, what you'll need to answer: flashy or fruity, whichever, and it's aggregate. It's a multiple fruit. Sorry, it's a multiple. So flashy and the aggregate. Multiple. Sorry. Once again, didn't wake up yet. 3 hours of sleep does not help me. Anyway, so you'll see the pineapple. You're saying it's a flashy and it's a multiple fruit. Done. Okay. Now the dry one. Dry, and that's a crepe myrtles, of course, from my front yard. Need to cut a couple of those trees down. But anyway, so in this particular case, what we'll see, the fruit, the dry fruit, which is automatically simple. So we have a dry fruit. When it's ready, it opens up, releasing the seeds. So it's a dehiscent fruit. Okay. Dry will be automatically simple, and the type will be dehiscent. You need to know that. Dry, dehiscent. Same thing with the cotton. That was a part of the fruit, the capsule. Whenever the cotton is ready, it will open up, releasing the seed, and we're using those particular fibers to create clothes, right? And the seed will be somewhere tiny, little bitty. Cotton seed will be in the middle, and later on we are removing the seed, squishing it to squeeze the cotton seed oil out of it. Again, peanuts, whenever it's ready, it will kind of open up at the seams, releasing the seeds. So all of them are dehiscent. All right, now acorn from my main neighbor's tree. Acorns. So right here, we're looking at whenever the acorn fall off, that particular fruit part will not open up, releasing the seeds. So that would be indehiscent. All right. Same thing with the sunflower seeds. Whenever they come out as the sunflower seeds. So another indehiscent. You don't really have to worry about the Samara or any of those. Don't worry the capsules. You don't have to worry about that. So yeah, so acne, yeah, don't have to worry about that as long as you see it dry, dehiscent or indehiscent. Is it opened up, or if it stays closed, it's indehiscent. So here we have the legumes, right? So in this particular case, opens up at the seams, releasing the seeds. Here, the seed stays within the wing. Wing takes it with the wind, can take it away from the parent plant, but it's a indehiscent seed. All right. So here we have some of the answers for you. All right. Now seeds. Whenever you're looking at the seed, this is the seed which is actually, well, if you can turn it, but basically whenever you open up, first of all, monocot or dicot. See, that particular round thing of the seed is the cotyledon, and that one is another cotyledon. So we have two cotyledons, it would be a dicot. Now, you will need to draw and label. So what you do is this. Actually, I think, yes, we're looking at the embryo. You see the embryo right here. And the embryo will have several major parts. The things which will turn in the actual leaves, then the stem, and then of course the embryonic root. And that's basically plumule, radical, and hypocotyl. I think, yeah, here we go. I did a really good job hand painting those ones for you. The green one, the green part right here, the leafy part, see right here, that part. But the leafy part is known as the plumule. It will turn into the actual leaves. All right. Then the blue part, the end of it, it's known as a radical. Radical will turn into a root. Then the red one, known as the hypocotyl, and hypocotyl is the stem. And of course, those particular things, well, this is a covering for the seed. So remember, seed will have always three parts: the embryo, the food source, and the colony. What's the difference between the angiosperms and gymnosperms? There is a question on the test about that. Fruits, the presence of fruits. The seed of the angiosperms is enclosed within the ovary, which generally gives us a fruit. All right. How many cotyledons do you find in the bean seed? And again, all of that is a food source and the sperm, essentially, cotyledons. 2 of them. What is the function of the cotyledons? I just told you it's a food source. For what? For the embryo. What part of embryo develops into a root system? Root radical, the blue part right here. So here, once again, we're looking at it's a dissecting scope, what you will see with your naked eye, essentially. The cotyledons, purple, the green one would be plumule, the red one would be hypocotyl, and the blue one would be a radical. Yep, all that stuff. So now we're looking at the microscope or cross-section of the monocot. Now let's kind of take a look what exactly we're looking at. All of that, all of this right here, all of light bluish color on the left, all of that is the endosperm, starch. Now cotyledon, here, all of that, all this, that part right here, between the endosperm and the actual embryo, that's the cotyledon. Here's the cotyledon, all this. Again, between the endosperm and the embryo. Now, radical and plumule. Radical, remember, root part. Plumule is the leafy part. Does it have the stem? Remember, well, think about the grass. Grass does not really have much of the stem. So that's why we're not talking about hypocotyl. So, but you definitely need to be able to recognize under the microscope, well, on the lab practical, you're coming in, there will be an arrow pointing. Name the part of the structure, and you're like, oh, that's the leaves, the top part, so that would be a plumule. And yeah, I will not flip them over. And then what this is the bottom part, so that's probably a radical. If it's pointing right here, it's a cotyledon. If it's pointing somewhere in here or several arrows pointing right there, that's the endosperm. So cotyledon, got it, radical, got it, plumule, got it, endosperm, got it. What is the function of the endosperm? Food source for the embryo, right? Already talked about that. Plumule of the embryo develops into leaves. How many cotyledons corn seed has? Only one cotyledon. Based on the number of cotyledon, can you predict maize is a monocot or dicot? Well, because it's a one cotyledon, it would be a monocot. Now that model, yeah, and here's the explanation. This particular model, you specifically looking, well, there is several of them. Here we're looking at the monocot, here we're looking at the dicot, and there is a little bit of difference between two of those. Don't worry about that part. So here's a monocot, here's a dicot. So whenever the dicot seed is growing, so see, this is the cotyledons, this is the first true leaves. Whenever it's growing, it will open up and the seed will literally come out from the ground. As a matter of fact, it will come out and it will open up with first two things. Those things are actually cotyledons. They will feed the growing seedling with whatever the nutrients they have, but they also will start photosynthesize. So those ones are cotyledons. So cotyledons help with the initial feeding of the seedling, then wither away and fall off. And by that time, we have a first true leaves appearing, and they will pick up the photosynthesis and so on and so forth. So the first part right here is actually cotyledons. In the monocots, what will happen? The seed stays inside of the ground in the soil, and the first thing which comes out is the actual true leaf. But look, here we have one leaf coming out, monocot, quite often, and then whenever you'll see two leaves coming out, that will most likely will be a dicot. But again, that's the actual leaves. The cotyledons are embryonic ones, the one which we are looking at, the one which carries essentially or helping with the nutrient digestion. Okay, so make sure that you will be able to see, well, the taproot, the main root, and then you don't really see the taproot right here. So what kind of root will have here? A fibrous root. What type of roots you see on the model? Taproot or fibrous root? Here we're looking at the fibrous roots. That one, and that one are taproots, because you can clearly see the main root with the secondary roots coming out. Here, you kind of hard to say which one is the main root. Make sense? All right, let's take a look. Closer look on that one. So again, we're looking at the seed staying in the ground. The first thing which coming out for the photosynthesis is the actual leaf, and the root system is relatively hard to identify which one is the main root. That's the monocot. And this is what the monocot, I think that's a dicot, preserved dicot picture. Now, here's what we're looking at whenever we're looking at the dicot, you'll start seeing that the embryos start growing, the root is going, you'll see the main root, secondary root coming out. Now the hypocotyl now opens up, and whenever it's kind of opens up, it brings out the cotyledons, the seed actually, out of the ground. And the cotyledons will open up and they'll start photosynthesizing. And then we'll see the actual true leaves coming out. So cotyledons right here. I think I have a different picture, kind of from the top. Yeah, here we go. So here, so you'll see that they are cotyledons right here, and they look different than the actual first true leaves. Okay. And I think I have the actual pictures later on. So here's the cucumber. That's the cucumber growing on the so you can see that those ones are nice and round. That's the cotyledons. And here we're looking at the first true leaves coming out. All right, seed dispersal. Some of the students have a lot of issues with the seed dispersal because they don't really understand how it works. On a lab practical, I'll try to keep an open mind. So basically, if you have a spikes of some sort, it's most likely will stick to the fur of the animal or a bird, and that would be most likely the animal dispersal. Then any kind of berries, the one which, well, the seed covered with the fleshy stuff, the animal eats the soft tissue, the seed goes through the digestive tract, comes out with the feces unharmed. The seed can start growing. That's also animal dispersal. So if you'll see a soft tissue surrounding the seed or some sort of spikes on the seed, that's most likely the animal dispersal. Now, wind dispersal, you'll have wings, so to speak. It will be a very large surface area, so the wind can pick it up and they will glide. They will not really fly, but glide away with the wind. Okay, now the water. If the water have to pick up the seed, so typically it will have a very thick layer, and I'll show you what do I mean by the very thick layer. So here we have air. So the seed itself, or wind dispersal, seed itself, and now we have the whole wing, creating a very large surface area, so the wind can easily pick it up, and usually they stay relatively small. Wind can pick it up and take it away. Cotton, again, huge surface area whenever it will live, and the seeds is embedded deep in there, so it will pick it up and kind of roll it away on the ground. That's a wind dispersal. Same thing right here. Oops, I guess I need to lower it up. So here we're looking at the dandelion seeds, right? The seed and huge surface area, so the wind can pick it up relatively easily. Now look at this one right here. First of all, a very thick layer with seeds, but also has some spikes. So technically it can be used by the animal or could be carried by the water. I have a thick skin, so to speak. Same thing here. You can see the spikes, which might get attached to this to the fur, or also it might be going by the water. So let's say if you'll see this on the lab practical, and you will answer water. Well, okay, I'll say, okay, I can see that the water surface area, spikes kind of floating on top. Or you will say animals. I'll see spikes, fine, I'll be accepting that particular answer. But if you'll say for that particular relatively big seed, air, I probably will not be accepting that one. Make sense? So I'll be fairly flexible if it's obvious, like this one. If you'll say air or wind, not so much. No, that's actually an animal. The animals eat the soft tissues, the seeds are coming out through the digestive system, tada, spread around. Now on those ones, see the spikes? Most likely attach itself to the fur. Therefore, most likely animals. What about this one? Well, again, we're looking at the berries, so animals eat them, the seeds will not be digested, so that's an animal. And I think, yes, seed, and you can put several of those ones, whatever we use, the cotton, wind, the any kind of berries would be the tomatoes, would be the animal. So dandelions, wind, sweet gum, that one known as a sweet gum, could be air, could be wind, could be animals. Here we have some of the wind ones, so it's up to you what you will put in the dispersal mechanism. Just kind of understand the logic behind it. Make sense? All right, and post our lab review questions. We are doing fairly well. What is the critical innovations made Angiosperms most successful group? Flower and fruits. What is the difference between pollination and fertilization? Pollination is transferring of pollen to the, or bringing the pollen to the carpel or pistil, technically stigma. And the fertilization is when the sperm fusing with the egg. A mature ovary becomes a fruit. After fertilization, mature ovule becomes a seed. What is the tissue represent in the seed that nourishes developing embryo? Endosperm. What is the name of the fruit made from the ovaries or many ovaries? Aggregate fruit. What is the difference between dehiscent and indehiscent fruit? Are those fruits fleshy or dry? They all dry, or both of them are dry. Dehiscent, whenever the fruit is ripe, it will open up, releasing the seed. Indehiscent will stay closed. How can you tell a monocot from a dicot? Number of cotyledons, 1 for monocot, 2 for dicot, but also leaf venation, right? Parallel for the monocots, typically speaking, and the something else such as palmate or pinnate venation, that would be for dicot. Root systems. If you'll see the taproot with the secondary roots coming out, that's most likely a dicot. Think about the roots which we are eating. Carrots. Clearly a taproot. So it's clearly a dicot. All right. And also the location of the vascular bundles. We'll talk about that a little bit over. And then of course, the flower parts, 3 multiples of 3 in the monocot and the multiples of 4 or 5 in the dicot. And reflection questions. Yep. Well, we are done with the lab. Let me